Oh, okay, that turns off for that. That makes sense. All right, 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay. Actually, I'll put this one on. All right, cool. Well, let's get started. So, good morning, everyone. So, hopefully you all know me from PHAR3320. I did a small little lecture series during that. And today, for the next, I think, 6 lectures, we're going to kind of continue those kind of themes that we're looking at in PHAR3310. So we're going to talk about neuropharmacology again. So central nervous system is going to come into play again. We're going to talk about the brain a lot. And specifically, we're going to get into a bit more of the nitty-gritty into some neurological diseases and disorders. So we're going to talk about depression and kind of antidepressants. We're going to talk about anxiety and hypnotics. We're going to talk about, what else are we talking about? Dementia and Alzheimer's. And then Dr. Alex Tang will finish up the lecture series with 2 lectures on photopharmacology and I can't remember the other one. Is it cannabinoids, possibly? I'm not too sure. So today's just going to serve as like a bit of an introduction to neuropharmacology. I know we talked a bit about neuropharmacology in some sense with the last lecture series looking at glutamate, GABA, dopamine, membrane transporters. So today's just going to be a bit more of a recap and kind of some of the drug targets that we can affect with pharmacology in the nervous system. So what do I want you to get out of this lecture? Again, we're just going to have a bit of a recap of PHAR3310, just so we're all on the same page with the central nervous system. Then we're going to talk about different drug targets at the synapse. So specifically, we're going to talk about ion channels, receptors, enzymes, and transporters. Yeah?
You never released those slides.
I put them up last night. Are they not there, or? Okay. I thought I did. Hold on 1 second. I'll just take this out so it's easier. In unit readings, is it, or no, lecture notes? Week 10 and 11. Items hidden from students. Okay, 1 second. Okay, yeah, this should be made available to you now. It was just hidden from you, and I thought it was automatic that it would show itself. Yeah, all good? Awesome. I will give just a little preface as well, is that in that folder there's about 6, all the lecture contents up there. I haven't updated a lot of those, so The night before each lecture, I'll update the lecture slides, so just make sure that you're using the most current ones. All right, so what we're going to talk about, again, is just so you can understand how drugs can affect the nervous system. We're going to talk a bit about ion channels, receptors, enzyme, and transporters. Then we're going to focus a little bit on the role of the blood-brain barrier and how that's important for neuropharmacology. And then we'll briefly touch on drug tolerance and dependence, and then we'll finish up by talking about toxins. And one of the ones I want you to focus on is botulinum toxin or Botox. All right, so first few slides, we're just going to have a bit of a recap of PHAR3320, just so we're all on the same page here. So does everyone know what this pink organ is here in the head? Yep, I'm seeing some smiles, I'm seeing some nods. That's great. All right, so that's the brain. We all have a brain. Arguably, I'd say the brain is the most important part of your body, but to each their own. So what is the brain? Well, the brain is the giant supercomputer in your head that controls everything you do, in a nutshell. So every thought that you have, whether that's conscious, unconscious, like me moving my hands right now, me moving my lips, my tongue, me pushing air out of my mouth, me breathing, my heart beating, that's all controlled by your brain. And it's happening in millisecond time scale that you're able to do all these complex functions. You, using your eyes, looking at these slides, hearing things, processing it as soon as I say it, this is all done by your brain. It's an amazing organ, and I really love studying the brain because there's so much we know about the brain, but there's so much we don't know about the brain. Anyway, I'll stop talking about that now. But when we're talking about neuropharmacology, we're talking about pharmacology or how drugs interact with the nervous system, okay? And the nervous system can be broken down into kind of two categories. You've got the central nervous system and the peripheral nervous system. So the central nervous system is what we refer to as the brain and spinal cord. And this is what I'm most interested in, mostly the brain. But there's also the peripheral nervous system, which is all the other parts of the body that's controlled by nerves and neurons that isn't the brain and spinal cord. So all the nerves, that innervate your skeletal muscles, innervate your GI tract, your heart beating, all that kind of stuff, that's all controlled by your peripheral nervous system. And we're not really going to touch on that in this lecture series. Mostly we're just going to focus on central nervous system and how drugs affect the brain. So if we take a slice of the brain, so let's just say we just cut, so you're looking at me now, if I just make a cut this way, and you're looking straight into my brain through my eyes, this is what you'll probably see. This is all being colored, different false colors just to show you different parts of the brain. But basically the brain's not a homogeneous structure, it's very heterogeneous. It's not just, one, like you see in this picture, it's just like a pink organ. The brain's very, very complex, and it's made up of many different structures and just many different tissue and cell types as well. And this is just showing you some of this complicatedness. So I don't know if you can see this square here with the brain and the slice. That's showing you where the slice is coming from, so it's about midway through the brain. And you can see all these folds, it's a 3D structure, it's all folded in on itself. You've got these sulci and gyri, you've got the cortex here, you've got different parts of the cortex as well. So the cortex is like this outer kind of layers of the brain, and then you got everything below the cortex, all the subcortical areas, and different parts of your brain control different functions or are important for different functions. So for example, this part might be important for more like motor control, so like me moving my hands and my mouth, whereas this part over here might be more important for things like cognition or sensory processing. But then you've also got all these subcortical areas that are all equally important. There's all different nuclei in there, there's different tracts, and a lot of these functions, so even though this might be, controlling your motor movement, that goes through some of these different structures here, so like the thalamus, different nuclei, cerebellum. So even though we can divide the brain into different areas, just note that it's very complex, and when we're talking about the way that drugs affect things, it doesn't just affect, like one specific part of the brain, it's going to affect globally different populations of cells and regions in the brain. And this, again, is just demonstrating some more complexity here. So this is looking at this one structure here, the hippocampus, which is important for things like learning and memory. But look at that hippocampus. It's also divided into different regions as well. So you've got the dentate gyrus, you've got CA1, CA2, CA3, and these are all very important for different functions as well. And if you zoom in even more detail, so you go 60 times, 100 times magnification, you're going to see little cells everywhere. And those little cells are completely different as well. It's not just completely homogeneous. So don't worry too much about all this information in different brain regions. I'm just trying to point at that the brain is very complex, and there's a lot of different regions and kind of neurons and cell types that we need to be thinking about when we're talking about neuropharmacology. And then just as an aside, this is a cross-section of the spinal cord. So if you just cut the spinal cord, this is what you'll see. And this just goes to show that the spinal cord is also complex as well. You've got white matter, you've got grey matter. So it's made up of a lot of tracts, but it's made up of a lot of cell types. And any drug that affects the brain can also affect the spinal cord as well. So it's just another thing to keep in mind as well. But don't worry too much about it. So again, just following on with the recap. So why is the brain able to be this supercomputer? How is it able to process all this information and control me waving my arms, talking like this? Well, that's because you have specialized cells in the central nervous system called neurons. Okay, we showed this before in PHAR3320. So just recapping. So these are neurons. This is your stereotypical looking neuron. And again, these are just the cells of the nervous system that generate those electrical signals and then send those electrical signals throughout the brain and to the body to control everything you're doing. And again, this is happening millisecond time scales. So sometimes you're not even thinking about doing something and your neurons are just firing constantly as well. But these neurons only make about 50% of the cells in the central nervous system. There's also other cell types in there as well called glia, which are like the supporting cells. So you might hear things like astrocytes that we talked about with glutamate reuptake. There's microglia, satellite cells, oligodendrocytes, all these other cell types. They also can be affected by drugs as well, but we won't get into too much detail with that. So just to reiterate what parts of the neuron we're going to be kind of looking and talking about. So these kind of finger-like projections and little knobbly bits coming out of the cell body are dendrites. So these dendrites actually, make some of the connections between other neurons. We usually say that the dendrites receive input. Then we also have a cell body. So you should all know what a cell body is. It's got a plasma membrane, it's got cytoplasm. It houses the nucleus. Within the cell body, you've got all your organelles as well. So mitochondria, Golgi apparatus, everything like that. So that's similar in neurons to other cell types. But one other striking change in these neurons is this kind of long projection that comes out of the cell body called the axon. And this axon is where those electrical signals are generated, but then where they also connect with other neurons on their dendrites to send that information as well. And these axons can be very small, so maybe they're just making a connection with a neighboring neuron, but maybe some of these axons are millimeters, or in humans, maybe even centimeters long, because they make such long projections to other neurons. And then at the end of the axons, you have axon terminals. So this is, even though the axon makes connections with other neurons, it's at these axon terminals where the actual kind of connection is made. So what you typically see in the brain is that you have one neuron connecting to another neuron. This is very simplified. In reality, there's probably tens, hundreds, maybe even thousands of neurons that is kind of connected in a network. But this is how we usually delineate neural transmission. So one neuron is here, one neuron's here. The space between the neurons, so the area between the axon terminal and the dendrite, is called the synapse or synapse. And this is where the space between the neuron is, and this is where kind of electrochemical signaling happens to transmit that electrical signal from one neuron to the other. And this is just demonstrating it here. So these dendrites, if you zoom in really high, they got these little projections coming off them called dendritic spines, and this is what actually makes the connection with the dendrites. And when we delineate these neurons, usually when there's a neuron connected to another, we say that one neuron is the presynaptic neuron, so it's the neuron before the synapse, and then one neuron is the postsynaptic neuron, so the part of the neuron after that synapse. So we usually say that the postsynaptic side is the dendritic spines, and the presynaptic side is the axon terminal. Okay? And then just going over neuron to neuron communication. So what happens is that this neuron will receive stimulation in some capacity. So that could be a stretch response, it could be a light evoked response, it could be electrochemical signals of some kind. And what happens is that that signal is going to open or close ion channels, okay? So you've all know what ions are, I'm pretty sure. So it's just like a charged particle. So things like sodium has a charge of +1, calcium has a charge of +2, potassium has a charge of +1, chloride has a charge of -1. So all those ions move into and out of neurons. And when you have movement of ions, you're generating electrical charge. That's a current that's moving in or out of the neurons, which generates a voltage. So when that stimulation happens, it changes those ion channels. Ion channels open or close. You get current moving in or out of the neuron. And sometimes when that current gets so big, you can actually generate an electrical response called an action potential. And then that action potential then generates from this axon initial segment down the axon. Once this reaches the axon terminal releases neurotransmitters, so those chemical signaling that we talked about in the first semester. And then these are able to affect their receptors on the postsynaptic side. And then if this, depending on what the neurotransmitter is, this might then go on to open more ion channels to then cause another action potential or electrical response, which then propagates as well. So again, this is just demonstrating one neuron to another neuron, but in reality, you know, there's tens, hundreds of neurons, and this is happening all at the same time. Some of these neurons cause excitation, some of these neurons cause inhibition and stop action potentials. And the brain relies on this balance between excitation and inhibition for normal functioning. Okay, does anyone have any questions? Again, this is just like a basic recap. Feel free to put your hand up, ask questions whenever. I don't mind if you butt in. So just following on in PHAR3320, again, we talked a little bit about ion channels, ligand and voltage gated. We talked about action potentials. We talked about transporters, passive transport, primary, secondary, active transport, and we talked about some different neurotransmitters and enzymes. But one last thing I just want to re-emphasize again is just the idea of this action potential, so the electrical signal that occurs. So this is just showing you the membrane potential, so basically the voltage of the neuron. Neurons usually sit pretty negative at about -70 millivolts, but then what happens when you get stimulation like neurotransmitters and things like that, it starts to open ion channels, and those ion channels allow ions to move in, which generates a current. So you start seeing, if you get more positive ions moving into the neuron, that brings that potential a little bit more positive. So it goes up to -55, for example. And if that stimulation is enough, so enough current gets through, you get this action potential, which is an all or none response. You get this firing until it reaches about +30 millivolts, and then you get kind of some of the ion channels turn off and some other channels open, and basically this brings the resting membrane potential down again. Don't worry too much about the details, but just know that this is the kind of action potential that we see that is responsible for that electrical activity. And then just looking at the synapse again, just so you can get a better look at neurotransmitter release. So you have an action potential coming down this neuron. This is the postsynaptic neuron's axon terminal. Electrical signal comes down, and then when this electrical signal comes down, you get calcium influx. This causes vesicles of neurotransmitters to dock to the presynaptic membrane, and then this exercitizes those neurotransmitters, and then those neurotransmitters are able to bind to their postsynaptic receptors or even presynaptic receptors, which we talked about autoreception a little bit. But that's it for the recap. So I know a lot of information we just went through really quickly. Hopefully you're all up to speed now, remembering from PHAR3310. But that's really what I want to just kind of emphasize, is just neurons communicate with each other. It's depending on this current and ion channel movement. And obviously neurotransmitters are released which can affect postsynaptic or presynaptic targets. All right, so now we're going to move on to the actual more neuropharmacology side of things. So just a few umbrella terms to start with. So what is neuropharmacology? Well, in a nutshell, it's the effect of drugs on the nervous system. So how do drugs affect the nervous system, the brain, spinal cord, and even peripheral nerves? What is a drug? So we're probably going here with the Rang-and-Dale pharmacology definition, which is just a chemical substance when administered to a living organism produces a biological effect. Okay? Drugs can be synthetic in nature, so we can make drugs that affect different endogenous receptors or proteins. They can be naturally occurring, they can be plants, they can be toxins, anything that just affects, that is administered and affects our body. You can have kind of endogenous drugs, so your body might be able to produce its own molecules and substances which affect receptors, but this isn't the drugs that we're talking about. We're talking about drugs that you have to ingest or is administered to your body. They might act on the same receptors as those endogenous molecules and drugs, but for a drug to be a drug, you have to administer it. And there's different levels of neuropharmacology. So we can talk about molecular pharmacology, and this is kind of at the level of the single cell. So basically, if you get a microscope, zoom all the way in, you're just looking at one neuron or two neurons. That's what we call molecular pharmacology. So you're just looking at how does a drug affect one population of neurons, how does a drug affect a specific protein on a neuron or ion channels. That's what will be relevant to a molecular neuropharmacologist. But then some neuropharmacologists might be behavioral neuropharmacologists. So this is looking at the behavior or level of behavior, which is the output of many neurons, circuits or brain regions. And then similarly, psychopharmacology is looking at the level of mood and cognition, which is the output of entire circuits and brains. So I'm going to talk about function a lot in the next few lectures, but just know that function is context specific. So for example, to a molecular neuropharmacologist, if I'm talking about function, maybe I'm talking about what is the activity of a single neuron, or what is the result of blocking an ion channel or opening an ion channel, looking at that kind of single cell molecular level. Whereas if I'm a behavioral neuropharmacologist, if you say function, I'm thinking about, okay, what is actually happening to entire brain regions that's kind of causing a response. So just keep that in the back of your mind. It's just a little bit context dependent, but I will probably use the word function a lot. And just one other thing to keep in mind, again, I talked about it at the start, but the nervous system, it's comprised of many different cell types. It's not just two neurons and a synapse, okay? Millions, trillions of neurons, different cells as well. Glia, some of the neurons are excitatory, inhibitory, whatever. There's a lot there, but just, you don't need to know about all the intricacies, but just keep in mind that, it's a bit more complex than what I'm going to be explaining here. So, neuropharmacology. So, obviously, there's different drugs out there. I know in pharmacology, you've probably covered a lot of different drugs and how they affect our body and different cellular systems. And just like there's many different classes of drugs that can affect the systemic body, there's many classes of drugs that can affect the brain and cause different responses depending on what targets they affect. So, we're going to talk about a few of these classes actually. So this is just a list showing you some of the different ones. So you could have, drugs like anesthetic agents. So these are drugs that are used to produce, things like surgical anesthesia. Examples of this, you might have heard of isoflurane before, which is a common anesthetic. There's analgesic drugs which are used for controlling pain. So things like opiates, anxiolytics, which reduce anxious-like symptoms. We're going to talk about that in the anxiety lecture or anxiolytic lecture, but things like benzodiazepines we'll talk about a lot. Then you've got things like anti-epileptic drugs, antipsychotic drugs, antidepressant drugs. So there's all these different classes of drugs that can affect neurons, but as you will see in this lecture series, it's not just cut and dry. So for example, an antidepressant drug, it's used to alleviate the symptoms of depressive illnesses. But many antidepressant drugs are also anxiolytics as well, because they work on kind of similar pathways that are kind of affected in anxiety disorders and depression. So you'll see kind of like a lot of crossover with a lot of these drugs. They're going to kind of coalesce on similar mechanisms. So even though they're delineated, they're different, some of these drugs might have kind of crossover or cross-modality effects as well. And when these drugs affect neurons, we usually say that they're modulating neurons in some way, so they're changing the activity of these neurons or changing their function. So we usually say that they're kind of neuromodulators in some way. Not all the time, but usually we do. And it's just important to keep in mind that some of these drugs can affect, this or modulate neurons on different time scales. So for example, if a drug, is really quick and it affects on the millisecond time scale, usually that will affect something like voltage-gated ion channels or ligand-gated ion channels, that are important for that action potential and activity. Some of these drugs might work on things like a second time scale, so it might affect things like slow synaptic transmission, which might be more of like G protein coupled responses. Sometimes they might affect the brain for hours or even days, so they might induce things like synaptic plasticity. So I don't know if you've heard of synaptic plasticity before, but basically it means that the synapses change. So some of the communication between the neurons becomes stronger or less stronger, so that affects kind of the output, because if you have a stronger synapse, you're going to get more activity, whereas if you have a weaker synapse, you get less activity. So some drugs can affect that synaptic plasticity. But you can also have some drugs that, if you take them long enough, they can actually induce changes for months to a year. And we'll talk a little bit about that with dependence and tolerance, but usually these drugs, if you take them for a long period of time, they can induce structural remodeling. So what do we mean by that? It means that we can actually change the way that the brain is shaped. So some of the neurons can make different connections to other neurons. Maybe they can lose connections, and that can also affect the output as well. So don't worry too much about these examples, but just know that these drugs, they can affect anything from millisecond time scale to days, months, even years, depending on how long you take them for. All right, so moving on to the drug targets. So how do these drugs that we're going to be talking about affect the nervous system? So let's just say you take a specific drug, what's it going to do to your body, or what's it going to do to your brain? There's a lot of different things it can do to your brain. Today we're just going to mainly touch on kind of like 4 targets, which are kind of the most common. And one of these is affecting ion channels, okay? So I already talked about it. Ion channels allow for the movement of ions into and out of membranes that generates a current that's going to cause action potentials, okay? So you can have, ion channels, they have their own kind of gating mechanism. So we talked about gating in the first semester, but what we mean by a gating mechanism is that these ion channels open in a specific way. So for example, some of these ion channels might open when a ligand binds to it, whereas some of these ion channels might only open when there's a change in that resting membrane potential. So when you start getting current going through the neurons and its voltage increases or decreases, then those ion channels can open or close. So usually say there's 2 types, voltage and ligand gated. And drugs can affect ions mostly in the sense that they can be blockers. So this is just showing you an ion channel here that's in the plasma membrane, which is this yellow part. So you've got an ion channel sitting in there. When an ion channel is open, ions are able to flow in and out of the neuron going through the membrane. But some drugs can actually be a blocker for this. So a drug can actually occupy that pore site. So even though the ion channel might be open in a sense, it's kind of blocked by this drug. So ions aren't able to move in or out. And this is really true for a lot of toxins that do this. They kind of block those ion channels. But you can also have drugs that are modulators of ion channels. So a modulator is a little bit different from a blocker. It doesn't actually occupy the pore. What it does is that it affects the ion channel indirectly. So it alters the opening and closing probability of these ion channels. So it makes it more likely to open or less likely to open, which will affect the amount of ions going through. So again, just different mechanisms here. So you can have blockers or modulators. And modulators usually affect things like G protein coupled responses, and then those go on to affect the ion channels either by phosphorylating proteins or things like that. So other drug targets that we can have are receptors. I'm sure you've all talked about receptors a lot throughout your pharmacology degree. I don't have to explain this to you, I suppose, but basically with receptors you can have agonists, which are substances that activate the receptor, or antagonists that bind to the receptor but don't activate it. So these drugs can be agonists or these drugs can be antagonists. When a drug binds to, or when an agonist induces its effect, for neurons it can do things like directly open ion channels or close ion channels. So again, affecting those ligand-gated ion channels, which will affect communication. But then they can also bind to G protein coupled receptors as well and cause a variety of different transduction mechanisms. So you can, the binding of the drug can change enzymes, it can modulate ion channels, it can even cause DNA transcription, which will go on to cause different proteins being translated, things like that. What other drugs affect the nervous system? Well, drugs can actually affect enzymes as well. So again, enzymes are important for breaking down molecules. So for example, you might see an enzyme like acetylcholinesterase, which breaks down acetylcholine, or monoamine oxidase, which we'll talk about later, which breaks down monoamines. So things like monoamine oxidase is really important for breaking down monoamines like serotonin, norepinephrine, and dopamine. So if you have drugs that affect these enzymes, either by inhibiting them or competing for them, you're going to affect the amount or levels of these neurotransmitters like these monoamines, which can go on to change neuronal activity and functioning as well. And then lastly, you can also affect transporters. So we talked about transporters before. They're proteins that move solutes either with their concentration gradient or against their concentration gradient into or out of the cell. In many cases, they rely on energy to move the substrate across, but like we talked about before, you can also have secondary active transport, which can get around this. And these transporters play an important role in the reuptake of neurotransmitters at the synapse. So again, neurotransmitters like excitatory or inhibitory, glutamate GABA, dopamine, acetylcholine. So if you block these transporters from working, you leave those neurotransmitters in the synapse for longer, which can cause a prolonged effect as well. And this is just putting it all together. So this is just showing you just kind of a more magnified view of the actual synapse. So these are vesicles with neurotransmitters. These neurotransmitters are released into the synapse, and then obviously there's all these different drug targets here. So you've got ion channels, you got ligand-gated ion channels or receptors. These neurotransmitters can bind to it to cause a postsynaptic effect. You can even have transporters here on the presynaptic membrane, so these neurotransmitters might be reuptaken through these transporters. But then you've also got enzymes here that can cleave. I've drawn it here as a Pac-Man. They can cleave these neurotransmitters to break them down, or maybe they interact with the molecules to produce a new substance. So again, just showing you that at this synapse, drugs can target many different proteins, receptors, ion channels, whatever it is. And then again, I keep hammering this, but just to reiterate it again, these drugs are also present on other CNS cell types. So it's not just this neuron-to-neuron communication. There's also those 50% of other cells like astrocytes, microglia, that have their own targets that drugs can affect as well. So it's easy to just look at what does this drug do to this one particular neuron. There's also other neural subtypes and glia as well. So it becomes really complex when you're talking about drugs and even just talking about the nervous system in general. And I showed this in PHAR3320, but this is just to reiterate that even on these neurons, so this is one neuron here, cell body, dendrites, axon. It's just showing you that there's many different subtypes of ion channels and ligand-gated ion channels and different receptors along these individual neurons as well. So just adding another degree of complexity. So for example, Kv2.1, this is a voltage-gated potassium channel 2.1. A lot of it's expressed here at the cell body, whereas potassium channel 4.2, I think it is, it's more expressed around these dendrites, whereas voltage-gated potassium channel 1 is more expressed at the axon. So you can have a drug that might be very selective and it only binds to Kv1 rather than Kv4, but you might have a drug that's kind of less selective and it affects all voltage-gated potassium channels. So again, we'll talk about it in the other lectures, but it's just adding a bit more complexity in that some of these drugs can be really selective, but they can have a very global effect as well, even at the level of the neuron. So yeah, very complex. And then also other drugs, so we talked about these targets. We won't talk about this today, but it's also worth mentioning that other drugs can actually target DNA and RNA as well. So you can cause more transcription, more translation. They can cause kind of genetic changes, mutations, things like that as well. But we're not going to talk about that. Okay. So a few other things I want to touch on before the end of the lecture is how do drugs actually get into the brain? Okay, so let's just say you take an oral drug or intravenous administration of a drug or gaseous administration. How does it get to the brain? Obviously, you have a circulatory system, vascular system. It moves through your blood, but it's not just that. It's not that simple. it's not just as soon as you take something, it's going to get into the brain. The brain actually has its own kind of, what would you call it? Kind of like its own protection called the blood-brain barrier. And you've probably heard of it referred to as the BBB. So neurons in the brain, they're very metabolically active, and they're in a very like equilibrium state, and they're very sensitive to kind of changes in the environment as well. So a change in the concentration of ions might cause neurons to just go really crazy and start firing. So the brain needs its own kind of protection to ensure that the environment has the correct concentration of ions, proteins, and that everything that's circulating in the body, maybe that's not healthy for the brain. So it needs a way to kind of filter these proteins and solutes out. And it does this through the blood-brain barrier. So this, in a sense, this blood-brain barrier isolates the nervous system from the circulatory body. It still allows oxygen to exchange, but it only allows certain molecules and ions to pass through. And the way it does this is just showing you in the diagram here. So you have central nervous system endothelial cells, which make up kind of the vascular capillaries in the cell, in the brain. The brain's very, 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 very vascularized. There's hundreds of thousands of capillaries everywhere, very fine ones. You have arteries as well, arterioles, but there's so many capillaries because your brain is so metabolically active. It needs a lot of oxygen. It needs a lot of kind of waste removal as well. So we have a really big circulatory system in the brain. And it's made up of these endothelial cells. These endothelial cells are in contact with a pericyte here. And then what you have is, I talked about those glia before, so astrocytes. And these astrocytes have their end feet, which kind of envelop these capillaries. And what these do is that these end feet create this kind of barrier that only allows certain solutes to pass through the cells or into the neurons. These pericytes, they have their own kind of function as well. They can actually restrict or open these capillaries as well, depending on the environmental needs. And they can also change the kind of gene expression of these endothelial cells as well. So they can enact some changes to kind of change this blood-brain barrier. But these astrocytes are really important for creating that barrier and maintaining it as well. So how do drugs move across the blood-brain barrier? This is just a diagram showing you it here. So you've got the endothelial cells with red blood cells in them. You've got your pericyte. Then you have your astrocytes, which are creating those that tight protection around it. And then in between these kind of spaces, you have these very small tight junctions. So I talked before that some gases can just move from the blood through the endothelial cells into the neurons. So things like carbon dioxide and oxygen just move through diffusion, just simple diffusion, that's fine. But then some of the other bigger solutes or molecules, they actually need to move through kind of mediated transport systems. So you can have carrier-mediated transport, which is seen here. And this is similar to those membrane transporters that we talked about. So they just get kind of transported down their, or up their concentration or down their concentration gradient with these transporters that physically bring them across. So the solute will bind to this transporter, and then this will get pumped into the endothelial cells and then into the neurons. But then you also have receptor-mediated transport as well, so where these neurons actually, you've probably heard of it referred to as like kind of clathrin-mediated transport, possibly. I don't know if you've heard of that. But basically at the membrane of these endothelial cells, you have these kind of dimples. And what happens is that these solutes go to the dimples and they kind of get encapsulated by those dimples. There's a lot of different kind of proteins there. It kind of snares them. And then that kind of endositizes that solute and then brings it through the cell. So kind of two different mechanisms there. So carrier-mediated, which is kind of more your standard membrane transporter, and then your receptor-mediated one where it gets engulfed and endositized through to the neurons. Not every solute or drug in the world has receptors or is able to move across the blood-brain barrier, and that's a real big kind of issue that drug development has, is how to, because maybe you can inject, like say you're using a mouse model, you can inject drugs into the brain and see an effect, But how do we actually get that into a human if it can't cross the blood-brain barrier? And that's where there's a lot of drug discovery there on finding ways to allow drugs to move across into the brain to have a therapeutic effect. Okay, so moving on, we're just going to talk quickly about drug dependence. And this is something that we might talk about in greater detail as we get to some of the other neurological diseases and disorders as well. But it's important to note that many drugs can affect the brain over a long-term period. So obviously if you're taking a drug for days, weeks, months, you can cause some changes in the brain, and some of these changes might lead to drug dependence. So this is where a drug is taken due to compulsion rather than a physiological need. Mostly you see this with recreational drugs. You also see this with a lot of painkillers as well, things like oxycodone. This, again, commonly results from repeated administration, and usually it's associated with the kind of the psychological effects of the drug. So it creates this desire to experience the effect of the drug. And this is in effect addictive behavior that can negatively impact the quality of life for the individual and those around them. Obviously, if they're drug seeking, then it can affect their health. But not only that, kind of the behavioral responses, but a lot of these drugs, if you use them for a long period of time or abuse them, you can, cause things like liver damage, you can cause impaired cognition, you can have a multitude of different side effects with drug dependence. And this is just showing you just a table of some of these drugs. So opioids, which we talked about, are used for analgesia, so pain relief. A lot of these can create very strong dependence. So things like morphine, heroin as well, oxycodone, that some of these might be prescribed to manage pain, but they can also result in some maladaptive changes, and one of these can be drug dependence. And again, like I said, it's mostly commonly associated with psychoactive drugs, or drugs that alter the reward system in some way. Again, things like heroin can affect the reward system. Things like recreational drugs like cocaine can affect the reward system as well, so that can create drug dependence. And over prolonged use of these drugs, why it does induce dependence is because it alters the ion channels and receptors and their functioning, and then induces long-term changes in neuroplasticity. So it changes the brain either at the synapse, or changes their connections, or maybe changes the proteins a little bit, so that it causes this drug dependence as well. We're not going to talk too much about it, but just be aware that it can do that. Now, there's several pharmacological strategies that can use to treat drug dependence, and this usually goes hand in hand with behavioral or psychological treatments as well, to kind of do it on a pharmacological level and also psychological level. And here's a table showing you. So it's just saying to alleviate withdrawal symptoms, for example, you can use methadone, which is used to blunt opioid withdrawal. So you can use therapies that block the effect of the drugs. You can use aversive therapies, so disulfiram. Usually this is used with alcohol dependence, and basically what happens is that you take this drug, and then whenever you drink alcohol you get a really, really sick feeling. You start vomiting and you feel nauseous. So it's basically using this drug to create like a negative association with the dependence that you had. So there's all different strategies to do this. And just like there's dependence, there's also another phenomenon called tolerance, and this is where you see a decrease in the pharmacological effect of a drug following repeated administration. Again, this often occurs with drugs of abuse, but not always. And this is just showing you kind of a particular dose response curve. So usually if you take a drug, for the first time, whether that's a painkiller or whatever, you need a certain amount of a dose to get a specific effect, and usually this caps out at a maximal point. But if you keep taking those drugs for a long period of time, you develop a bit of tolerance, so that all of a sudden that little bit of drug dose you needed doesn't actually elicit a response anymore. You get a rightward shift. So you need more of the drug dose to get that small effect, and then when you want that maximal effect, you need even more amounts of that drug. So that's just showing you that some of these drugs you can develop tolerance to. And tolerance can occur, again, because of plasticity in some regards, but it can also work because your body adapts and kind of metabolizes the drug a bit more efficiently, so then you need more of the dose to cause a response as well. But this happens a lot with a lot of the drugs that we're going to talk about, especially with depression and anxiety as well. All right, and then to finish off, I just want to touch on one or two slides on toxins as well. So we talked a lot about drugs, and mostly we're thinking about drugs maybe in more of a therapeutic sense, but toxins are also drugs that can affect the nervous system. So evolution has provided, plants and animals with the ability to produce toxins. Usually these toxins are used as like a self-defense or predatory mechanism. Now, a lot of toxins actually modify ion channel flow and the opening and closing of these ion channels. Many of these natural toxins are made to, again, incapacitate prey or defend against predators. But a lot of these toxins, the way that they work is that they work on these ion channels. So usually they block the pore of the ion channels, like we talked about in the start. So you stop ions moving in or out of neurons. And they can also interact with these ion channels to change the conformation of them. So you can cause them to open or close or reduce their probability to do so. And this is a list of some different toxins. You'll see that a lot of these toxins are in aquatic animals, but not all of them. So you've got a spider here. And there's all different toxins. One of them I want to drag your attention to is tetrodotoxin, which is seen in fish and octopus. And this affects voltage-gated sodium channels. So if you're exposed to this toxin, it actually blocks those voltage-gated sodium channels. And like we talked about earlier, those channels are really important for neuron communication. So that's why this specific toxin causes paralysis. So it can depress breathing, it can depress movement, it can depress thought, because your neurons aren't firing those action potentials. So if, for example, your throat or your lungs aren't getting those action potentials, then it's not going to work. You become paralyzed, or you can even stop breathing and die. And those of you who like watching Simpsons, I grew up on The Simpsons. One of the early seasons, Homer goes to eat blowfish in one episode, which has tetrodotoxin. It's called fugu. He refers to it as fugu a lot. But this is basically what's happening, is that the reason why he nearly dies in that episode is because it's thought that he's ingested pufferfish that had tetrodotoxin. You can eat pufferfish, but specific parts of it you can't, which is like the liver, things like that, because there's a high degree of tetrodotoxin. But just like animals and plants produce toxins, also bacteria produce toxins as well. So you might have heard of diphtheria before, which is a bacterial toxin. One of the more popular ones that you probably hear about is botulinum toxin, or Botox. Botox is one of the most potent neurotoxins that we have on the earth, but we use it for cosmetic surgery or therapeutic surgery as well. And the way that botulinum toxin works is it actually affects the motor neurons, okay? So let's just say you are exposed to Botox or botulinum toxin. The botulinum toxin goes into the motor neurons. So these are the neurons that control skeletal muscle contraction. And then what it does is that it stops these motor neurons from releasing neurotransmitters. So they're not able to stimulate contraction of those neurons anymore. It effectively paralyzes that skeletal muscle. So that's why we use it for cosmetic surgery a lot, because, for example, let's just say you inject Botox into your forehead. When you now scrunch up your forehead, you're not going to see those lines anymore because those muscles are effectively paralyzed. They can't contract anymore. Whoa, that was weird. And you might have also heard of tetanus toxin as well. So we always get vaccines against tetanus toxin. This is another toxin. I think it's present a lot in soils and things like rusty nails or metal, things like that. Tetanus toxin works similarly, so it affects those motor neurons, but it actually transports all the way back to the inhibitory neuron. So the inhibitory neuron basically causes inhibition on the motor neurons, so it causes them to relax. But if you stop that inhibitory neuron from releasing those neurotransmitters on the motor neuron, that motor neuron is now not getting any of those inhibitory signals, so it's unable to relax. So you get constant contraction, or tetanus, which you might have heard of. And a lot of these toxins are lethal in very, very, very small quantities as well. So it just goes to show you that Botox, even though we use it and it's kind of seen as like, it's very safe to use, it's actually one of the most potent neurotoxins, and if it's not administered properly, it can result in some bad side effects. But don't let that scare you off getting Botox if you want to get it. And that's it, really. So neuropharmacology, again, we talk a lot about neuropharmacology in kind of therapeutic contexts, so things like, improving depressive symptoms, anxiety symptoms, things like that. But neuropharmacology is really, really, really useful to just basic neuroscience. So how does how does the brain work? How does plasticity occur? How does one region talk to another region? We can exploit these drugs if we know what kind of targets or receptors or ion channels they work of. And then if we have like an experiment, for example, we can start applying those drugs and kind of look at what that does to the body. So we can learn so much about our own brain by exploiting these drugs as well. It doesn't always have to be for a therapeutic or recreational context. Anyway, so that's it. So thank you very much for attending today. Again, if you have any questions, I'll wait around. But yeah, I'll post the I'll post the lecture notes every night just before the lecture. And I'll see you tomorrow, same time, same place, for the depression lecture.